transmission started from the bat coronavirus to the Manis pendata and Homo sapiens. Homo sapiens means the human species. So there are a lot of SARS variants already reported, different disease and different virus which is responsible for this SARS disease. So the HCOV229E was reported long back, and in 2011 the Middle East, the MERS COVID variant was reported with the basic reproduction number 0.7 and case fatality rate of 4. 4 means 4% of the people infected are died. With the symptoms like pneumonia, sore throat, fever, cough, chills, and dyspnea. So that was the bat camelis vector host. That was considered as the intermediate host. And then comes the SARS-CoV, that is with the basic reproduction number 3 and CFR number 0.1. So that produced respiratory distress, fever, dry cough, headache, chills, and myalgia. So, and the devastating infection with the COVID-19 virus, that is SARS-CoV-2 virus, that started in 2019. The theory bats to pangolins transmission was still under question mark, and many people have ignored that theory. So they produced pneumonia, ARDS, fibrosis, fever, dry cough, and coagulopathy. So these all conditions. So people are confused. So what to do? They are running for the oxygen cylinders and medications. The vaccination camp is going on. So everywhere in a crisis. So this is the general pathway of COVID-19 replication. The SARS-CoV virus 2. It has the spike proteins attached to the cytoplasm of the cell membrane, and they will enter inside the host cell, and release of nCoV genome, that is ssRNA. ssRNA strand will be released. Then replicase and translation of viral polyprotein, and it is a complex structure. Replication transcription complex is formed with the RNA genome and genome with and subgenomes like RNA, sgRNA, all these happened, and the nucleic acid material genome RNA replication with the sgRNA transcription, the nucleocapsid N, the spike S1 and S2, membrane M, envelope E. So all these things happened within the vector body, and transition of viral structural proteins, the S, M, and E proteins at the EPR membrane. It is attached to the membrane of the endosome, and the COVID genome is introduced. So the S, E, and M protein combine with the nucleic capsid, and finally the assembly of a mature virion. That means the COVID inside the Golgi vesicle, and it will be released by means of coughing. It will spread into the atmosphere. And main important proteins, the mainly 4 proteins: the spike protein S1 and S2, nucleocapsid N, the membrane. Hello.
आपसे सोचने की
अच्छा
मैं सोच रही थी पर आप भी थोड़ा बिजी लग रहा है
नहीं मैडम, ऐसा कोई बिजी का वो नहीं है। आप बताएं
Hello.
कैसे घर पे सब बढ़िया?
बैठिए ना मैं
नहीं देखा था मैंने, आप
Hello.
नीचे शायद ऑफिस में थे।
Hello, sir. Somebody is speaking. Can you please?
Sorry, sir. Sorry for the trouble. Sorry, sir. Sorry for the trouble.
Okay. It's okay. I'll continue. So there are different proteins, 5 proteins, the S1, S2, and the nucleocapsid N, the membrane M protein, and envelope proteins with the ssRNA. And this is the main site of attachment of the COVID virus into the surfaces. That means there is a receptor binding domain of the angiotensin converting enzyme 2 type of receptor that is available with the spike proteins. So many people targeting on this particular area, that is the angiotensin converting enzyme 2 receptor binding domain for the vaccine as well as the drugs development. So once this happened, people started testing to find out whether the persons are infected with the virus or not. So we have heard about a lot of testing methods: RT-PCR, RT-LAM, CRISPR, Cas2, enzyme-linked immunoassays, rapid diagnostic test. And nowadays, everybody is believing on the RT-PCR test only that will detect the N gene, E gene, and the RNA-dependent RNA polymerase. So sample will be taken from the nasopharyngeal swab or esophageal swab. So time point will be the symptom onset. So after the onset, the RT-PCR will be giving positive. So which will have, and all these tests have difference in the reliability, then the detection, limit of detection, low sensitivity, low selectivity, high specificity, high reliability based upon the various techniques used in the COVID testing. And symptoms. So there are common symptoms like fever, dry cough, tiredness, and less common symptoms like aches and pains, sore throat, diarrhea, conjunctivitis, headache, loss of taste or smell, skin rashes, discoloration of fingers or toes, and serious symptoms like difficulty in breathing, shortness of breath, chest pain or pressure, loss of speech or movement. So this will, the person to person, the symptoms will vary. So the person who is infected with the virus will have either of any of these symptoms. So this is the infectious versus days after symptoms onset. So 0th day is considered as the symptoms will appear. Before that, actually the persons will be infected, and it will reach the peak at somewhere around the first day, the 0th before the 0th day. So this is the curve. This is a normal distribution curve. It comes to a peak and then declines. And at the end of 7th day, the symptoms will be less. So now, based on this, the therapeutic strategies have been developed. So stage 1, early infection phase. So we have either no symptoms or mild symptoms, and clinical markers like lymphopenia, reduced CRP level, and increased interleukin 6, and viral burden will be very, very low. So these time no therapy is needed. So this is the therapeutic strategy. And second phase comes to the pulmonary phase. So slowly the dry coughing, fever, the the shortness of the breath, headache, and glass opacities observed in the CT scan reports, and mild hypoxic conditions are the clinical markers used. So viral burden will be intermediate and spread from the lower respiratory tract. And now the symptomatic treatment starts. And stage 3 is the hyperinflammation phase. That means it is the observed symptoms are pneumonia, chest pain, productive coughing, multiple organ failure, cytokine storm, ARDS, severe hypoxemia, acute kidney injury. That is the last stage. Kidney injury, kidney damage will happen. So the viral burden will be highest. So the expansion throughout the respiratory tract, and so we have to reduce the inflammation. Mechanical ventilation is required, and hemodialysis is also carried out on such patients. So day 5 and 10 are important. 5th day the maximum, and 10th day there is minimum. And this is the immune response. So initially the antibody production will be very low, and inflammatory leukocytes will predominate. There will be a lot of influx of CRP and interleukin 6. So initially antibody production will be low, and as time proceeds there will be more antibody production, and the person is sick. So the prevention guidelines, this is WHO given prevention guidelines: washing your hands with the detergent, sanitize your hands, no smoking, wear a mask. Now it has made into double mask in India. So avoid unnecessary traveling, stay home. Stay home has produced a lot of problems. The industrial sector, then social distancing, and main important thing is do not touch your face with your hands. So don't use your hands to touch your face. So W updates as on 2nd, it is actually 2nd July, it is not June. So except African region, new deaths continue to decrease. So in South African region, still the count is increasing, and all other places the count is slowly decreasing. Around 57,000 new deaths per day, and over 370,000 cases per day. So altogether 180 million cases, over 2.6 million new cases, and almost we have lost 4,000 human due to death and due to COVID. African region recorded a sharp increase in the incidence, around 33% increases at a mortality of around 42%. And WHO has given a concern for the variants alpha, beta, gamma, delta. Alpha is beta, that is B117, and beta is B1351, and gamma is P1, and delta is B1617.2. So now we shall move on to therapeutic strategies. So there are antiviral therapy. We have to reduce the lot of virus in the body, like the remdesivir, hydroxychloroquine, chloroquine, lopinavir, ritonavir, and ivermectin. Then we'll be using anti-SARS-CoV-2 neutralizing antibody products. So the covalescent plasma, the reagent COVID-2, that is a cocktail of casirivimab and imdevimab. And another cocktail has been using valbenibib and istesavimab. Various immunomodulatory agents have been tried: corticosteroids, interferon beta 1A, interleukin 1L, antagonistic like anakinra, and the interleukin 1/6 receptor monoclonal antibodies like cetuximab, tocilizumab, sarilumab, cetuximab. Then various Janus kinase inhibitors like baricitinib, ruxolitinib. Then Bruton's tyrosine kinase inhibitors like acalabrutinib, ibrutinib, lisabrutinib. Then suddenly the start of the post-COVID syndromes like mucormycosis, and people were running in search of a specific drug called amphotericin, that is liposomal amphotericin. So liposomal amphotericin is a nanotechnology product. Then the miscellaneous treatment like 2-deoxyglucose symptoms improvement, for symptoms improvement and spend less time receiving supplement oxygen. So apart from this therapy, the oxygen therapy, management of acute hypoxemic respiratory failure in COVID-19, high flow nasal cannula, and non-invasive positive pressure ventilation, NIPPV. So these are the oxygenation techniques used. So then comes to the vaccination. So we have studied in the microbiology a lot of vaccines, inactivated, live attenuated, subunits or any fragments or any special proteins, and viral vector, then DNA vaccines, mRNA vaccines, all these vaccines. So inactivated vaccines, the examples are Sinovac, Sinopharm. Then the live attenuated vaccines, Codagenix, Ankara, and subunits, Novavax and vaccine. Then viral vector vaccines like the Oxford and CanSino. Then DNA vaccines like Inovivo, Cytoscatillas vaccine. Then mRNA vaccines like Moderna, BioNTech. So there are so many companies are associated with vaccine manufacturing, and so many institutions are working in coordination with the industries to develop vaccines. And many countries, even India also, we have got 2 vaccines approved for emergency use. That is, one is Covishield and another one is Covaxin. So now we shall switch over to the nanotechnology perspective. So this is used as the future application of nanomedicine to mitigate COVID-19 pandemic. So a lot of research is going on, because nowadays the nanotechnology products are slowly started gaining the acceptance. Previously, people were a bit reluctant to accept it. Now, because of the COVID-19 pandemic and vaccine production, people started slowly accepting the nanotechnology concept of manufacturing various medications. The nanoparticle approaches presented here will help to win the fight against the SARS-CoV-2 and other pathogens. So we all know that nanoparticles are the particles in the range of nanometric, is from starting from 10 to 10,000 nanometers with unique properties, size, polarity, structural and chemical composition. So the nanotechnology can improve the effectiveness and specificity of drugs. So we can improve the effectiveness, the site-specific delivery systems for the nanoparticles, and specificity of drugs and vaccines with unique physicochemical properties of the nanoparticles, which makes them useful in making the drugs and vaccines. And main crisis is the polarity of the nanopolarity of the drug, because the drug should have a well-balanced water solubility and lipid solubility for oral absorption. But for getting a drugs to be effective, it has to be attached to the proteins, and the polarity of nanoparticles can be modified to facilitate binding with other drugs, increasing the surface stability, reducing aggregation, because the nanoparticles tend to aggregate. So it will reduce the aggregation and precipitation. So they are small in size, they can easily interact with biological target. And the nanoparticles can pass through several types of membranes and several junctions, the lung blood vessel junction and blood brain barrier. All these are specialized membranes. So it can pass through several types of membranes. So nanoparticles, we have these special metal nanoparticles like copper, silver, gold, the inorganic materials like graphene, silicon, the various protein, carbohydrates, lipids, RNA, DNA, or conjugates, polymeric nanoparticles. So a wide varieties of materials have been used to prepare the nanoparticles. So nanoparticles of copper, silver, gold, the graphene nanoparticles, and nanoparticles attached with the proteins, carbohydrates, lipids, the RNA, DNA, it can conjugate. The various polymers can be used to prepare the nanoparticles. So the nanoparticles enhance drug therapy by optimizing the drug intake, the nanoparticle intake, and stability also increased. And their main advantage is target cell specific delivery of nanoparticles. Even we can incorporate magnetic properties to many of the molecules, many of the nanotechnology products. So already we have seen how much devastating the morbidity conditions and mortality conditions worldwide the Corona-19 virus have spread. Scientific community is working hard to control the pandemic by diagnostics, drugs, vaccines, and devices. Nanotechnology approaches have high potential to enhance conventional diagnostics, drugs, and vaccines. Even various personal protective equipments, gene silencing, creating biosensors, developing pharmacologically active compounds, and nano vaccines for directly destroying SARS-CoV-2 viral particles. Even lipid nanoparticle mRNA vaccines, it is a lipid nanoparticle based vaccine, mRNA. mRNA vaccines are already in use. The Pfizer Moderna vaccines. So these nanoparticles directly interact with the virions and reversibly disrupt their structure and render a virus incapable of replicating within the host cells. So we are using a lot of protective equipments to fight against COVID. The doctors, the healthcare workers, the frontline workers, they are using a lot of materials. And now it has been slowly converted. So we'll be giving a special coating or special attachment of many impregnated nanoparticles like the coats, the scrubs used in hospitals, the surfaces of the various hospital beds were impregnated with the nanomaterials. So this can enhance the efficacy of personal protection equipment. Even hand sanitizers, air filter systems have been modified with the nanotechnology. The nanoparticles potentially fight against SARS-CoV-19 by enhancing biosensors and diagnostic tests, drug therapies, designing new delivery mechanisms, then optimizing vaccines. So this technology overcome the difficulties facing conventional medicines. Conventional medicines, there is a problem because once the medicine is entering into your body, so it will have a different, it has to reach the specific proteins. And before that, it will go everywhere. So we cannot target. Normal medication cannot be targeted. So we have to make a targeting type of nanoparticles with binding with some enzymes or similar materials to the nanoparticles will enhance the efficacy of these type of dosage forms. Nanotechnology will revolutionize drug-based medicines in the 21st century, and COVID-19 crisis sets the stage to evolve the concept of nanotechnology into a reality. Now the nanotechnology has become real. Around 50 formulations are approved. Nanotechnology products have been approved by FDA, and out of which few nanotechnology products for COVID-19 treatment, especially vaccines. Vaccines are more predominant. So there are two types of strategies. Physical prophylaxis. Physical prophylaxis means we have the enhanced PPE with virus bacteria killing capabilities. So we have many types of nanotechnology products, the carbon nanotubes, liposomes, gold nanoparticles, missiles, the graphene oxide, silver nanoparticles, iron oxide nanoparticle, copper oxide nanoparticle. So all these systems, so they are used for upgrade HVAC filters, heating, ventilation, and air conditioning filters. A lot of filters are used in the treatment areas, and the people where the patients have been treated when various the because almost everywhere the business is going to be standstill. The film cinema theaters, so even foreign countries, they have made the HVAC systems, and these type of nanomaterials increase the efficacy of commercial disinfectants and hand sanitizers, and they decontaminate the hospital beds, scrubs, white coats, and ventilators, and enhance PPE with virus bacteria killing capabilities. And next comes to the treatment strategies, the immunomodulation. So they remove the pro-inflammatory cytokines and chemokines. So they remove the interleukins, the other chemokines. So these nanoparticles create a nano vaccines or a substitute traditional adjuvant. So we can change the adjuvants. So nano adjuvants have been made into practice. So they increase the antigen searching leukocytes for immunity and optimize efficiency and safety treatments. So they will optimize the safety and efficacy of the treatment methods. So we have vaccines, nanotechnology based vaccines. We have nanotechnology based vaccine and ordinary vaccines. So nanotechnology, nanoparticles can biconjugate and genetically engineer and if infuse, mineralize, or even assist in self-assembly of viral and bacterial particles. So these techniques are used as tools following vaccine design and production. So various designs are available for nanoparticle-based peptide vaccines like adenovirus, class 1 dsDNA virus, adeno-associated virus, human papilloma virus, human immunodeficiency virus, and these can be modified into carriers for targeted gene protein delivery. So we are hearing about the mRNA-based vaccines, BNT162b2, used by the Pfizer BioNTech. BioNTech was the research firm, and Pfizer has got the capabilities to manufacture vaccines. So they reduce the risk of infection with the efficiency of 95%, they claim, after the second dose of mRNA-based vaccine, Pfizer vaccine. The nanoparticles that deliver the mRNA contain a polyethylene glycol, PEG. So normally they are pegylated. So there was an initial hiccup with the vaccine production because they have produced some severe allergy symptoms and they had to replace with this PEG. And similar case with the RNA-based vaccines, Moderna, mRNA 1273, which is also a lipid nanoparticle-based vaccine. So both were granted emergency use authorization by FDA. So next is Novavax NVX-CoV2373 vaccine, which is a recombinant nanoparticle-based vaccine. It is a recombinant technology-based vaccine, which incorporates a full trimeric spike glycoprotein with a saponin-based adjuvant. Here we are using the saponin-based adjuvant. The US biotechnology company Novavax protein-based COVID-19 vaccine is being locally manufactured by the Serum Institute of India under the brand name Covax, but they are under trial, especially the children below age of 12 years. But there is a problem with the trial also. The people who are reluctant to send their children, who want to take the risk. And in phase 1 to human clinical trials, the vaccine could elicit neutralizing antibodies like anti-spike immunoglobulin antibodies and specific T cells response. So Sinovac biotech company also designed a nano vaccine against SARS-CoV-2 and successfully tested in mice. So they incorporate receptor binding domain subunit of the spike protein combined with two adjuvants. Though they have used two adjuvant system, they wanted to incorporate the receptor binding domain. So RBD, now onwards we'll be discussing it as RBD domains. So it is a receptor binding domain subunits of spike protein combined with two adjuvants, the monophosphoryl lipid A, MPLA, and CGP oligonucleotides. CGP is short single-stranded synthetic DNA molecules, and CGP-ODN, which stimulate transmembrane proteins like TLR4 and TLR9. So these CPG-ODN, it is a short single-stranded synthetic DNA molecules that contain a cysteine triphosphate deoxynucleotide. And the vaccination of mice was achieved in 3 stages: original shot with 2 doses, and resulted in a potent and protective T cell response accomplished by neutralizing immunoglobulin A antibodies. And this nanovaccine is currently in clinical trial phase 3, and they have got the Sinovac, I think the US FDA has approved for emergency use. So this is off note. Another nanotechnology vaccine was recently found to induce persistent antibody production and long-lasting memory response for at least 7 months in mice. So this trial is going on. And that special technique in this vaccine design, the receptor binding domain with RBD of the spike protein was conjugated via a special technology called spike tag or spike catcher technology with ferrine nanoparticles. They have used ferrine nanoparticles for this. So the receptor binding domain of spike protein was conjugated with spike tag or spike catcher technology. Hence, the unique capabilities of nanoparticles could revolutionize the process of vaccine design, manufacture, and delivery. So there are a few more vaccines like the protein-based adenovirus vector vaccine AstraZeneca with efficacy 70%, Johnson & Johnson adenovirus vector vaccine to express SARS-CoV-2 spike protein in the host cells to induce immunity. So which yields the Johnson & Johnson vaccine yields the SARS-CoV-2 specific antibodies in 90% of the individuals after the first dose. So people are running behind the Johnson & Johnson vaccine, waiting for Johnson & Johnson to come over India. But it gives a rare. Yes.
What's that?
Somebody has.
Yeah. Sorry, sir. Sorry.
Okay. So better mute their mics.
Yeah, I am muting you all. So
Okay.
I can take you from first. Before that, we will
Okay. Yeah, I'll continue. The Johnson & Johnson vaccine has got a risk for cerebral venous sinus thrombosis. So this has been observed. So many people are having problem with the similar type of symptoms in vaccines. And mild to moderate symptoms like pain, swelling at the injection site, and systemic like fever, chill side effects are very common for current COVID-19 vaccines. So another design is vaccine design could combine layered double hydroxide nanoparticles. So it is a layered double hydroxide nanoparticles and a plasmid encoding short hairpin RNA, shRNA. shRNA is short hairpin RNA, the viral RNA. And vaccine design could combine layered double hydroxide LDH-NPs nanoparticles and plasmid encoding short hairpin RNA, that is shRNA. To silence the expression of targeted genes such as essential the SARS-CoV-2 proteins to stop infection early. So early infection can be stopped. The LDH nanoparticles are compatible with mammalian cell lines and can insulate the shRNA against degradation. Thus, it is giving a promising delivery mechanism. So nanoparticles can be engineered directly to target SARS-CoV-2, the immunomodulatory factors to prime and alarm the immune system. Once the immunomodulation makes the alarm, it will give the immune system. Reduce inflammatory responses during COVID-19. And even the nanoparticles have been used for drug delivery of many agents. All interfering RNAs against conserved regions of SARS-CoV-2 into lipid nanoparticle formulations. It has suppressed the viral replication and improved survival of infected mice. So this is under the preclinical level only. Studies are going on in animal studies. Then graphene oxide nanoparticles, GONPs. So these graphene oxide nanoparticles increase the leukocyte numbers. So they will increase the leukocyte numbers such as macrophages and T cells. They are immune. They will increases the immunity. So this affects boosts adaptive immunity in individuals, and there will be a better immune response and viral clearance. So these graphene oxide nanoparticles are used as vaccine adjuvants. Even nanodiamonds, the carbon nanoparticles, elicit an anti-inflammatory state in the macrophages. So they will produce anti-inflammatory state. And repurposed carbon graphene sheets remove pro-inflammatory cytokines and cytokines from the blood of patients. So these are the techniques, fewer techniques to remove the pro-inflammatory cytokines and interleukins from blood of patients. And major problems of vaccines and medications will be overcome by nanotechnology solutions. Like many drugs are sensitive to acidity, water insolubility, absorption. So all these problems, the water insolubility problem can be overcome by nanotechnology, sensitivity, acid, base. Then the absorption problems, all these problems can be overcome by the nanotechnology solutions. Nanoparticles can increase drug delivery efficiency, binding to encapsulating hydrophobic or pH sensitive drugs. So hydrophobic drugs or pH sensitive drugs can be bound with the nanoparticles and creating a targeted release. So we can target the medication. The nanoparticles bound to drugs modified using organic molecules that provide better release characteristics. So we can attach drugs with modified with nanoparticle bound drugs with using organic molecules like cholesterol. Cholesterol can be used to modify the drug release profile of hydroxychloroquine. So other nanoparticles can facilitate the transport of 2 or more drugs. Suppose if you want to administer 1 or 2 or 3 drugs, then we can make use of the nanotechnology principle so that the dose can be reduced. Like the augmented, the combined outcome. So the combined outcome will be more additive effects of 2 drugs with less dose is possible with nanoparticles. The spike catcher spike tag technology, that is a new technology, allows irreversible conjugation of a recombinant protein by adding sequence of the spike tag peptide, 13 amino acid units. This peptide is having 13 amino acid unit, the spike tag, to its DNA sequence. So this spike tag spontaneously react with the spike catcher protein. So this spike tag technology product will spontaneously react with spike catcher protein and allows oligomerization. So this system was employed to generate mosaic nanoparticles that display multivalent antigens of SARS-CoV-2 spike RBD with along with other RBDs from different animal beta coronavirus to enhance beta cell responses, and they elicit high titers of cross-reactive neutralizing antibody. So they give neutralizing antibody content. So enhanced titers. So already we have discussed some of the techniques to reduce the secondary infection. The physical barriers used, then surfaces and air filters, sterilization of surfaces and air filters by antimicrobial nanoparticles, and sanitizers and disinfectants. The personal protective equipments like PPEs, gowns, scrubs, white coats, contact surfaces. So the masks and gloves are upgraded with nanoparticles of antimicrobial and antiviral capabilities. So the nanoparticles are there. These substances are coated with a mixture of nanoparticles to protect healthy and infected individuals. So cotton fabrics can be enriched. The fibers, cotton fibers will be enriched with zinc oxide nanoflowers to trap and denature SARS-CoV-2 spike proteins. So it will denature the spike proteins. So cotton fabrics enriched with zinc oxide nanoflowers. Then iron oxide nanoparticles and silver nanoparticles neutralize various strains of influenza and coronavirus and physically binding to SARS-CoV-2 virion by preventing internalization into host cells. Oxide nanoparticles. So they are having antimicrobial capabilities against respiratory tract pathogens. So the respiratory pathogens, they will have an antimicrobial activity against the copper oxide nanoparticles. So the problems in nanomedicine delivery, most of the drugs are having poor cellular uptake and insufficient stability under physiological conditions, and they will be unstable under different pH. So our body is comprising of different compartments and compartments having different pH. The non-target effects, that means the drug will affect the other targets which is not meant to affect. And sometimes the drugs will produce excessive immunogenicity. So that is also a problem. And these problems can overcome by short interactive RNA molecules bound to nanoparticles. So these RNA can be bound to the nanoparticles. And they can interact with a biomarker on desired cell population. So this is used as a biomarker and interact with biomarkers of desired cell population by localizing the drug's effects to avoid unnecessary contact with other cells. So drug localization attained. Once the drug localization attained, the drug will not come in contact with the other cells and reduces overall toxicity. So examples are nanoparticles coated with specific antibodies for especially the spike proteins, cellular receptor SARS-CoV-2 spike proteins. So that is an elegant delivery system for any drug that requires cell specificity. Many help to reduce the dose of medication. So dose of medication can be reduced and off-target side effects also reduced. So there are a lot of techniques: polymers, dendrimers, quantum dots, nanobots, microscopic robots. So these are the new technologies developed, but they are slowly coming into the play. In future, we will be using such type of technologies for treatment. The quantum dots, the arrays are used. Even the gold, silver, copper, they are precious metals. So by reducing the size, that means in the nanoscale, we can save the precious elements. The next is the amphotericin B liposomal novel drug delivery system. So we have seen the mucormycosis condition after the COVID infection. Many patients were reported with serious mucormycosis, and many people have lost their eyes. And the drug of choice was the amphotericin B, which is an antifungal medicine. But everybody is running for the amphotericin B liposomal novel drug delivery system. So complexing amphotericin B with lipid structures almost avoids the negative side effects of amphotericin. So main problem with amphotericin B is the nephrotoxicity. The nephrotoxicity of amphotericin B can be reduced by formulating in terms of lipid nanoparticles. So next is another technology, TechClo. It is a patented technology. That means hypochlorous acid is used. That is why it is, the name indicates TechClo, a reactive oxygen species. This hypochlorous acid is a reactive oxygen species naturally produced by the neutrophils of human immune system to fight against infections. So it is a reactive oxygen species which will fight against the natural, fight against infection. So HClO is in aqueous, it is in the aqueous solution to be inhaled and reduces viral load in the upper respiratory airways in recently infected individuals. So the Applied Pharma Research Company, they have got the patented for patent technology TechCo. So APR AOS 2020, that is a product, is designed to cleanse the nasal passage with hypochlorous acid, destroying the COVID-19 virus. Suppose the SARS-CoV-2 viral load in the upper respiratory airways is lower, it is likely that the probability of virus to enter the lower airways, damaging lungs, can be reduced. So it is an adjunct therapy using hypochlorous acid, HClO, which is a reactive oxygen species which naturally produces by the neutrophils in human immune system to fight against infection. So that can be delivered in the form of a nanotechnology product, the APR AOS 2020, thereby reducing the virus load in the upper respiratory tract. So we will administer this particular medication in the form of inhalation. So that will reduce the lower airways damage. So early and next is in the diagnosis. So we will be using a lot of diagnostic tests to detect the SARS-CoV-2 virus or COVID-19. and rapid detection is key factor. So we have to so test. That is a government policy. Then go for the treatment regime. So early and rapid detection is key for lowering the basic reproduction number of infected individuals. The nanoparticles can be engineered as biosensors for biomarkers, for detection of biomarkers, including the nucleic acid. Nucleic acid, DNA, RNA can be detected, or specific antigens. So we'll be using biosensors to sense or detect biomarkers like DNA, RNA, specific antigen with specific proteins, enzymes, or antibodies. So there are a lot of these type of antibodies. The DNA are the proteins, enzymes, and accurately detect SARS-CoV-2 virus. And recent advancement in nanotechnology can or allowed for release of SARS-CoV-2 detection platform that uses graphene conjugated anti-spike antibody. So graphene is a nanoparticle. Graphene is conjugated with anti-spikes antibody. So the recent advancement in nanotechnology used this graphene conjugated nanoparticles. So this novel kit requires no sample pre-treatment. So there is no pre-treatment required. The time consuming will be less. And labeling and impressively affect the detecting SARS-CoV-2 at very low concentration. At very low concentration levels of SARS-CoV antibodies can be detected by a specific kit. Sometimes alternative detection methods like dual functioning plasmonic biosensors. So these biosensors which tap into energetics of DNA RNA hybridization. This based on the DNA RNA hybridization, they will use the energetics of, and they will function as dual functioning as plasmonic biosensors. So there will be interaction with the graphene oxide particles and coated with fluorofin bound DNA target strands that can detect viral helices. So there is challenges and limitations of use of this nanotechnology in therapy. There is variability between results. So one research institution is performing a research and another one same research, but there is a lot of variability. So translating the efficacy of these particles from in vitro to in vivo situation is quite difficult. So it is really difficult to translate the in vitro techniques into in vivo situations because of the variability between the results. And large scale production of nanoparticles also very difficult because the large scale production of nanoparticles requires sophisticated instrumentation and the product cost becomes much higher compared to the normal conventional formulations. The required sophistication of manufacturing process of nanoparticles and intellectual property rights can drive up prices, the patent rights, intellectual property rights. So the entire health cost expenditure crosses. And another limitation is a risk of unwanted tissue interactions and toxicity. So already I mentioned the drug will be going into the other cells which is not required, the target sites, and that produces tissue interactions and toxicity. Similarly, the nanoparticles also will do similar interactions. So unwanted crossing of blood-brain barrier. Normally, blood-brain barrier is highly sophisticated. The capillary endothelium is acting as the barrier. So many drugs which will go through the pore mechanism, pore transport mechanism, because of the size. Next is accidental inhalation. There is possibility that these nanoparticles can be inhaled. So they will cause epithelial injury, pulmonary inflammation, and which contribute fibrosis, depending upon the size and chemical composition of nanoparticles. The size as well as chemical composition makes the nanoparticles toxic. One such example is epithelial injury by accidental inhalation of pulmonary for epithelial injury and pulmonary inflammation contribute fibrosis. Moreover, these nanoparticles interfere with biological process. So a lot of biological processes involved in COVID or COVID-19 infection, inflammation, oxidative stress, mitochondrial function, macrophage phagocytics and platelet functions. So they will modify, they will interfere with this biological process. The acute or chronic toxicity of nanoparticles by the generation of reactive oxygen species, cell membrane binding, DNA damage, altered cell cycle regulation. The entire cell cycle regulation is altered, or it will denature the proteins. Another important issue is to understand the long-term effects of nanoparticles in humans and the environment. So it is really difficult situation because a study with chronic administration of nanoparticles to rats resulted in structural damage in their testes. So including disorganization of spermatogenic cells and misoriented testis and reduction of germ cells. So they are unable to reproduce. So the importance issue like long-term effects of nanoparticles must be thoroughly studied in humans, in animals. So environmental scientists have to take care of this. Then allergic reactions and anaphylactics to the mRNA lipid nanoparticle vaccines. Already I have discussed this Moderna, Pfizer vaccine. So the design, the P-glycolated design. So these limitations and other unknown risks should be taken into consideration by evaluating actual potential of nanoparticles. So when we want to have the actual potential of nanoparticles, we have to consider all these limitations, the plus and minus of nanoparticle therapy. So now let me conclude. So the nanomedicine offers unique potentials to address the epidemiological changes with other emerging virus. So now the time has come. Lot of viruses is in the pipeline. We can say it is in the pipeline. Once SARS-CoV is there, there is a report of other viruses in different parts of the world. So viruses is in the pipeline and the medication is in the pipeline. The ongoing COVID-19 pandemic has shown that healthcare systems were underprepared. So our healthcare, so we have seen many countries, many developed countries were underprepared. The initial stages of COVID-19 pandemic. Many countries like US, France, Italy, they were underprepared to face the challenge, but slowly they have overcome the situation. So the current status of the field lends in favor of nanoparticles supporting modern medicine, but that but is important. Long-term side effects remain hard to assess. So we have to think about the long-term side effects that has to be studied. So a new discipline, medical science discipline, has come to emerge, which focuses on detecting and eliminating new viral or bacterial threats using nanomedicine and nanodrugs for diagnosis and therapy. Because new viral and bacterial threats are underway. So a new discipline of medicine that called theranostics for diagnosis and therapy based on nanomedicine. So this field, the nanotechnology field, has demonstrated futuristic applications such as spike protein specific nanoparticles, neutralizing nanomaterials. So all these comes up under play. So many people are, many industries, many organizations are doing research. So it may even become a pharmacological standard of care once the side effects are well understood and mitigated. While true benefit of nanomedicine in the fight against COVID remains to be seen, it is worthy of in-depth consideration and efforts. With these words, I conclude my session. Thank you.
Thank you, sir. Now the session is open for query. Anyone, any participants have doubts can ask now to sir.
Yes, sir. Hello.
Hello.
Ma'am,
First of
we
all,
can hear you, please.
Yes.
hello. Can you hear me?
Hello. Yes, yes, I can. I can hear.
First of all, thank you very much for your excellent compilation of all the information of COVID-19 and the nanoparticles. My question is that, according to you, so which is the better vaccine for children also and for the adults also?
See, now we
According,
have. Can I answer you?
yeah, yeah.
See, now people are working on the already this disease is only 1.5 years, not even 1.5 years, 1 year, 3 months, and people are trying with trying to develop vaccines under different schemes,
Yes.
nanotechnology based, RNA based, DNA based, the live vaccines, attenuated vaccines. Or every vaccine has its own merits and demerits. So time will tell. That's all I can say. Because all vaccines have one or other problems. But main thing is the immunogenicity. They will produce immune response. It should not overexpress. There is only one criteria. There is nothing like which vaccine is bad. Am I answered you?
Yes, sir. Okay.
Are you satisfied
Yes, sir.
with my answer?
Yes, sir.
Okay, thank you.
Okay, okay, sir. Anyone else? Anyone else?
Thank you, sir, for sharing most valuable informations about the relevant topic, sir. Really, it
Yes.
was a very wonderful session, sir. Thank you so much.
So you are a
Anil Kumar, sir,
speaker, you are a coordinator or Prasita?
no, she's Prasida is in the chair, sir, now.
Okay, 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 okay. Yeah, yeah, because
Uh,
yeah.
that has been showing in the YouTube. Now the video is streaming live in YouTube, and
Yeah.
some participants are watching in YouTube also.
Okay, okay, fine, fine, fine. That is very nice.
So thank you very much, sir, for coming to our program. Thank you so much.
So let me say thank note at the end.
Yeah, yes, sir.
So I thank the AICT FDA team, FDP team, which has given me an opportunity to speak in this platform. And I thank the principal of National College of Pharmacy, Dr. Sujit Verma. Thank you, sir. And the organizers of the adult FDP program, National College of Pharmacy. Thank you very much for your support in my presentation. And I thank the participants of this program. And thank you very much.
Okay, sir. Thank you. Thank you so much. Participants, please note that our next session will be on 1:30 p.m. The details will be uploaded in the WhatsApp group, in our WhatsApp group. So thank you all.
And thank you, Dr. Akash Marathakam. You are the coordinator, right?
Yes, sir. I am the coordinator, sir.
Yes, yes, yes. Very nice, very nice. Continue to do this such type of programs, so that will help a lot of people in the, especially the scientific communities and the teaching community, will be benefited out of such programs.
Okay, sir. Thank you. Thank you. With all
Thank
your
you.
blessings, we can do like this more, sir.
Definitely, definitely have to do.
Yes, sir.
We have
Yes,
to
sir.
continue with. So everywhere, almost all AICT approved institutions are doing such type of programs.
Yes, sir. Yes,
And this year also, we are also conducting some programs.
Yeah, I
The
know, sir.
program is going on right now from 5th to
Right now.
9th. Same
Yeah.
time, that's what we couldn't participate in your program. And you people also could have
Yes, sir.
participated in our program also. But
Okay, sir. I think another one is also there, sir, for your college. Another one FTP is also approved for.
yeah, one more is there. That I think you can, because you will be having another program, but plan accordingly that it should not coincide with our program, so that we can both community, the scientific community and teachers